My name is Isabel Gulings. I'm Scientific Director at ILSI Europe, and I'm thrilled to co-host this webinar together with John Norman from the American Chemists Council. The aim of this webinar is to discuss indeed the recent report published by the World Health Organizations, and we will elaborate on future challenges and the research needed in this area. But before starting our scientific program, allow us to briefly introduce both organizations who are co-hosting and co-organizing this webinar. ILSI Europe stands for the International Life Sciences, and we're the European branch of a global federation. Our mission is to provide guidance to the food sector through collaborative science. This guidance can touch upon topics within the field of food safety, nutrition, and sustainability and food security. Collaborative science is the cornerstone of everything that we do. And this collaboration is created between the public sector, industry companies, the public sector, but also academic experts. A clear example of this is, for instance, the Microplastics initiative that we have at ILSI Europe. And here you can see the composition of this task force that drives and proposes research activities that is then taken forward by expert groups, which contain more than 50% of academic experts. Our classical deliverables are peer-reviewed publications, but we also provide black and white documents, which are guiding documents for the industry, and also monographs, which can explain a very complex topic in a more easy to access document. Besides producing and assembling science and publishing it, we also spend a lot of time and dedicate to dissemination and communications of these deliverables, such as we have our own annual symposia, specific thematical symposia, and webinars at CIS, where we make sure that the science reaches the right audience. So this example today is a beautiful example of it. We do this together, this time with the American Chemists Council. So I'll give the word to my co-host, Norm, to introduce the organization.
Hi, my name is John Norman, or Norm. I am a senior director here at the American Chemistry Council in Regulatory and Scientific Affairs. We are part of an international effort at the International Council of Chemical Associations to develop a research program on microplastics science, primarily to develop quality data necessary to conduct a risk assessment on the environmental and potential human health effects of microplastics. This is just a brief schematic as to some of the projects that the American Chemistry Council and some of my colleagues are working on, just as an illustration to show the type of work that we're partnering with academic institutions on to try to generate and publish. So back over to you, Isabel.
Thank you. So now let's come down to the program that we have. We have a beautiful lineup of experts in this field, all contributed to the World Health Organization report. We will start with Todd, who will be introducing the report and the dietary exposure pathways, and also summarizing key aspects of the report itself. Followed by Professor Boobis, who will be more talking about the uptake of small particles in the gastrointestinal tract, so the toxicokinetics of microplastics. And then we will end the presentations with Dr. Stephanie Wright, who will talk about more the toxicity related to the dietary exposure and challenges linked to this. And we emphasize that we will, in this webinar, focus mainly on the dietary exposure and less on the inhalation. All speakers will indicate future research needs. In the end, John Norman will give us a wrap up, and he will be moderating the Q&A session at the end. So don't hesitate to continue while the speakers are presenting to put your questions down in the Q&A session box. So besides, I already mentioned this, the Q&A box you can find at the bottom of your screen. We mentioned that this webinar is recorded, and you can find it afterwards on the website of both organizations. Of course, feedback is a gift to us to improve for the next time. So please, you will receive after the webinar a feedback questionnaire, and we would gladly appreciate if you take a brief moment of your time to give us this feedback. So this in general, let's now start with the real content of the webinar. And the first speaker is Dr. Todd Gouin. Dr. Todd Gouin received his PhD specializing in the field of environmental chemistry from the Trent University in Canada. And then following his graduate studies, he obtained both experimental and modeling experience in assessing diverse chemical exposure, including current use of pesticides in Costa Rica, and also on polycyclic aromatic hydrocarbons in Arctic region in Alaska. So he has covered a round number of countries in the world. More recently, he has worked in developing and applications of tools aimed at both screening and prioritizing of chemicals and high-tier risk assessment methods for Unilever. And nowadays, he provides consults on a large range of cutting-edge topics, including the development and application of risk assessment methods for particles, such as microplastics. Over to you, Dr. Gouin, and thank you in advance for your presentation.
Thank you very much, Isabella. It's fantastic, and thanks for the opportunity to communicate output in relation to this report that we worked on behalf of the World Health Organization. And in this presentation, I'll just focus on one particular aspect of the report, which is related to the dietary exposure to nano and microplastic particles, with implications to trying to think about exposure in terms of human health. Oh, there we go. Just want to begin by giving a link to the report. If you haven't read it or seen it yet, I do encourage everyone to try to have a look. The report covers quite a number of aspects in relation to exposure to microplastic and the potential implications. And I think it's fair to say that in this session that we will cover, try to cover some of the highlights, but there are a lot of aspects that are sort of important to also reflect upon. In addition to Alan and Stephanie presenting here, I think it's also important to acknowledge other contributors to the report, which includes Fleming Cassie, Shirley Price, and Sandra Wagner, all of whom provided valuable input to the development of the report based on their individual expertise. And I also want to give acknowledgement to all of the peer reviewers, external peer reviewers, who provided valuable input to earlier versions of the report, which I think really helped to greatly improve the quality of the report and its direction that we work towards. Now, I'm going to assume that everybody probably knows who the World Health Organization are, WHO, and that WHO regularly issues health-based guidelines on health and the environment, and they also monitor, of course, emerging issues such as plastic, which has been identified as an important issue by the Health and Environment Department at WHO. And there have been two publications then now in relation to microplastic. There was one published in 2019 related to microplastic in drinking water, and then this most recent one that was published in this past August that tried to look at the human health implications from a broader perspective in terms of exposure, such as in relation to inhalation as well as water, but also other foods and beverages that may be of importance. In helping to support both of these reports, I think it's important to draw attention to a number of publications that provided a systematic review of the quality and reliability of data that have been published in relation to microplastic in the peer-reviewed space. These include a publication from Bart Coleman's and others in 2019, whereby they developed a set of criteria, quality assurance, quality control criteria to evaluate studies reporting on microplastic in fresh and drinking water, which was followed by a publication led by Stephanie Wright in 2021 in relation to evaluating the quality of studies reporting microplastic in air. And then most recently, a publication led by myself published last year related to evaluating the QA/QC of toxicological effects studies, both mammalian in vivo and in vitro studies in relation to exposure to microplastic. And Stephanie, I think, will discuss a little bit about that in her presentation. These systematic reviews, I think, are really important in helping to provide us a relative indication of what the potential implications are related to exposure to microplastic, but also are useful in helping to provide guidance towards recommendations on future research that could help to strengthen the overall quality and reliability of studies that are present in the literature. Now, I think one thing when we look at the literature is fairly clear is that the presence or the detection of microplastic has sort of been described as being ubiquitous. So we can find microplastic pretty much everywhere on the planet, which is perhaps not too surprising because wherever there are people, we tend to bring plastic with us. And so since plastic is also ubiquitous in the world, there is sort of this knock-on effect that microplastic is also occurring. Now, I think the leading theory around this is that we do have an unintentional release of plastic waste into the environment, which results in its fragmentation and degradation to form these microplastic particles. And one of the key challenges is trying to characterize and quantify the relative importance of these different emission sources in relation to where the particles get released in the environment, and then try to understand the fate and mobility of these materials that may eventually result in exposure either in the environment or in the context of these presentations in relation to human health. And so there's a lot of effort, I think, that has to go on towards trying to better understand the sources of these materials, because this is very useful for helping to provide guidance in terms of mitigating strategies that could be effective at trying to reduce our overall exposure to not only microplastic, but also try to prevent the release of plastic waste into the environment, which I think is a very important sort of goal that we should all be striving towards. Now, microplastic has been broadly defined as particles that are less than 5 millimeters in size. But I think it's important to be aware that these materials do reflect a heterogeneous mixture of particles with that varying shape and size and polymer composition associated with them. And depending on how they are used in commerce, how plastic might be used in commerce, for instance, they could have a wide variety of different types of plastic additive chemicals associated with them. This, combined with differences in surface chemistry, can all sort of have an potential impact on the knock-on effect of an adverse effect that might occur in a biological organism. So trying to better understand the complexity of materials that we might be exposed to is really critical for helping to guide an understanding of what factors are associated with these particles that might be resulting in an adverse effect. So I think there's some need to try to better refine our overall understanding of what these materials actually are in the surrounding environment that we might be exposed to. Now, this becomes somewhat important because when we think about our exposure from a human perspective, what we know about our exposure to nano and microplastic particles is that it is ubiquitous. So we know that these particles are in water, they're in air, they're in soil. And so when we look at the data that are available, it's clear that we have an exposure, particularly an oral exposure, for instance, of particles that are greater than 10 microns in size. And this has been shown to be associated with various foods, beverages, and drinking water. What we know we don't know a lot about is these particles that may have a greater physiological relevance to us. And these are particles that are less than 10 microns in size. And I think it's important to sort of differentiate what we know between particles greater or less than 10 microns, because from an adverse effect or from a human health implication perspective, it's important to understand the relative differences that how these particles behave once we ingest them and they enter into the digestive system, for instance. And I think Alan will sort of reflect upon why these particle size is sort of an important element to start with to try to better understand what the implications might be associated with this. Now, there have been a few studies, for instance, that have reported the clearance and elimination of microplastic in human stool. And these, again, are associated with fairly large particles that are greater than 10 microns. But what we would perhaps benefit from is a more mass balance understanding of what it is our ingestion looks like relative to what this clearance and elimination is for particles of varying size, so that we have a better understanding of what the potential accumulation of these particles might actually look like. So there's a lot of data needs, I think, that we need, particularly reflective of these smaller size particles. Now, thinking about the problem in a more broader context, where microplastic come from, this idea that these are particles less than 5 millimeters in size really sort of originates from a publication from Thompson et al. In 2004, where the term microplastic was coined. And one of the things in this publication that they draw attention to is sort of a need to establish whether there are any environmental consequences of this debris. Now, the focus of this particular publication was on plastic debris that was in the marine environment, which was degrading and fragmenting into microplastic particles. And the emphasis or the focus was really on what was happening to these materials in the marine environment. as a consequence of that, we really began sort of perhaps chasing the knowledge and having a bit of a reactionary response to the presence of plastic in the marine environment, and thinking about how filter feeders, for instance, might be exposed to these particles and potentially concentrate them in their tissues, which we then might ingest. And so there was a lot of emphasis and a lot of data reporting microplastic in seafood and fish that exist in the literature, which was also accompanied by a number of studies looking at microplastic in drinking water. So a real focus on the aquatic environment. But more recently, we become aware that not only is the aquatic environment contaminated, but so too is the air, soils, and we also become aware that perhaps contamination might be coming from food packaging. So our understanding is perhaps moving a little bit away from this reactionary response to hopefully becoming more proactive and trying to understand where the sources of contamination might be coming from. Now, this is an observation that's not too dissimilar to what EFSA were sort of advocating in their report of 2016, where they also identified a need to better understand the effects of food processing, for instance. They also recognized this shortcoming in the data. It reflected of a lack of understanding of these smaller sized particles. However, in the EFSA report, they focused on particles less than 150 microns, whereas here I'm sort of emphasizing on that 10 micron size range. But what we do know, because of this emphasis on seafood and fish, there have been a number of studies that have attempted to try to estimate what our daily per capita ingestion of these particles might be. So in this table, there's a number of studies that have reported on this, but I really want to draw attention to the size of particles that groups have been reporting on, and you'll see that these particle sizes tend to be much greater than 10 microns in size, and they really do reflect the larger size particles, which have a very different physiological response or potential physiological response in particles that might be less than 10 microns, where we really have no data and a clear lack of understanding of our exposure to these smaller size particles. So consistent with EFSA, there's sort of a need then for research that helps us to better understand smaller size particles, but also the implications with respect to toxicokinetics and toxicity. So sources of contamination, I think, do represent a really important research need in trying to move forward. We're clearly moving away from understanding that the environment might be contaminated with plastic and microplastic, and moving towards better understanding the implications that food processing and packaging may have in terms of impacting our potential exposure to nano and microplastic particles, but also considering the impact that atmospheric deposition might have. And so there's sort of a change or a shift in our understanding of where the contamination is coming from, which has implications for helping us to potentially improve our overall understanding of what the exposure might be. This is effectively sort of repeating that message in the slide, and also trying to draw attention to, I guess another of the limitations is that we have is that the types of foods and beverages that we've been looking at tend to be, again, following this reactionary response, where we have an understanding that sea salt, for instance, may be contaminated with microplastics. So we end up with a lot of studies reporting concentrations in sea salt. Some vegetation, such as honey, there has been speculation that could be contaminated because of atmospheric deposition onto flowers, which are then contaminating beehives. And then we have some studies on milk and beer, which have also received quite a bit of attention. And so what we're sort of seeing then is this need to try to better understand, again, that source of contamination relative to the types of foods that we're looking at. Now, underlying all of these data are concerns around the quality assurance and quality control that are associated with the monitoring data that we have, that we see in the literature. And one of the challenges is that when trying to report or monitor concentrations in a particular environmental matrix or food or beverage, is that there is a lack of standard methods that are available. This is sort of compounded by a challenge that a number of studies lack analytical verification, and that not all studies are actually verifying that the particles that they are identifying in their studies are actually microplastic, although they might be reporting them as microplastic. And then finally, there are concerns regarding this background contamination issue, again, related to the fact that microplastic are ubiquitous in the atmosphere. If you're working on collecting samples from the field or in the lab, there's always a potential for your samples to become contaminated. And so this all sort of confounds the challenge to characterize and quantify what our exposure might be. And so having a stronger adherence to quality assurance and quality control is really a key recommendation moving forward. So Based on this, we do see in the report a number of concerns regarding the representative of the quality and reliability of the data, but also in terms of the types of foods and beverages categories that have been studied to date. This is also echoed in a publication from Cox et al., who, when they evaluated the same type of literature that we did, they estimated that only approximately 15% of Americans' daily caloric intake was accounted for, which could account for a significant underrepresentation then in terms of the exposure. So it would be helpful to try to better understand what concentrations might be associated with more representative foods and beverages, but also whether or not there are cultural, socioeconomic, age, and gender differences that might be associated with exposure. So moving forward, there's a recommendation that we should perhaps develop a targeted approach, exposure assessment approach, not only sort of focusing on what is happening in the environment, but also considering the manufacturing process and whether or not there are steps in the processing and packaging stages where microplastic might potentially be introduced into the foods and beverages that are being manufactured. If we could identify these sources of contamination, there is a potential then for us to effectively mitigate those sources. But then there's also potential contamination associated with consumer use and habits. So what happens in a person's home may also represent a potential source of contamination, which would be beneficial to try to better understand. So overall, a holistic approach that considers the total food basket and which evaluates the potential sources throughout the whole life cycle of producing foods and beverages is sort of advocated in moving forward. So the key messages related to the exposure chapter in the report are that human exposure to both nano and microplastic particles are understood to be ubiquitous, and that this occurs by all exposure routes, and that information on exposure from air, drinking water, food, and beverages are generally limited. The data on the characteristics of microplastic and their quantification in all important media are necessary with a need to better understand their sources of contamination. Finally, research needs, sort of mentioned standard methods. There is a real need to try to develop standard analytical methods and sampling methods that can help us to better understand the contamination of foods and to characterize these particles in a robust way. Both of this really requires a need for us to have reference materials that are representative of environmentally relevant nano and microplastic particles. And then finally, a need to better understand the sources of contamination. Would greatly help to initiate effective strategies at mitigating exposure. And then finally, better understanding of uptake and fate for both the inhalation and oral ingestion of exposure with a better understanding of the toxicokinetics aspects, which is something that Alan will sort of continue on in his presentation. So thank you for your time. Hand it back over.
Thank you very much, Todd, for this first introduction and interesting presentation. You already gave the bridge to the next speaker, which is Professor Alan Boobis. And it's an honor to present him as well as Emeritus Professor of Toxicology at the Imperial College London, where he was a member for over 40 years. And his main research interests lie in mechanism toxicology, the mode of action, and chemical risk assessment. He has published over 250 original papers and is proud to have an H factor of 80. He is and has been a member of several national and international advisory committees, including current chair of the UK Committee on Toxicology, member of the WHO study group on tobacco product regulations, but also on JACFA boards and FAO and WHO GMPR, so pesticide residues, and is a member of the Board of Trustees of ILSI Europe. So we're really proud to have him on board and give us such a lovely advice each time. Professor Boobis, over to you to talk more about the toxicokinetics of microplastic.
Thank you, Isabel, and hopefully you can hear me.
We do hear you. Thank you. Everything is fine. You can.
Okay, thank you. And I'd like to thank ILSI Europe and ACC for the opportunity to participate in this webinar and present something on the toxicokinetics of microplastics. What do we know and what do we need to know and don't know at the moment? I should have control of this, but I don't seem to be. Yeah. So just to reiterate Todd's comments that this report, the WHO, was the work of a large number of individuals, not only the lead authors, but those who contributed to the expert consultation and reviewing, and I'd like to acknowledge their contributions. But whilst this presentation reflects the outcome of the WHO deliberations, the content is my own responsibility and reflects my own personal views, and so the WHO contributors shouldn't be held directly responsible. So following exposure, as Todd's outlined, to microplastics and nanoplastics, we have to consider what happens to them in the body. And the linkage between exposure and effect, which Steph Wright will talk about, is of course toxicokinetics. And toxicokinetics of anything, any chemical or any substance, is really important because it allows us to understand the dose-response relationships, and it contributes to the weight of evidence to understanding the plausibility that exposure results in adversity. And I think for something like an environmental exposure like microplastics, this is really important, that we can find the plastics, as you've heard, ubiquitously through the environment. And then the key question is, to what extent does that impact on human health? And how plausible is this given the uptake into the body? And so that means we need to understand the fate of the particle following exposure. As you've heard, we're going to focus on the oral route of exposure in this webinar. So following exposure via food or beverages, we have to consider absorption in the GI tract. And that which is absorbed is subject to distribution to the different tissues, where of course it may have an adverse effect. And then we have to consider, does it persist indefinitely in the body, or is there some mechanism for clearance and elimination, and how much of it will be eliminated over time? And I'd like to say right now that the vast majority of particles are too small, too big, I should say, to be eliminated by the kidney. The effective pore size of the kidney is such that the particles will not be cleared to any significant extent, except for the very smallest of particles. So in the GI tract, the microplastics have three possible ways of moving from the lumen into the systemic circulation, where there's the potential for an effect. And these are, they could migrate across the cell, so transcellular, the particle gets into the cell and then passes through the epithelial cells lining the gut and into the circulation. Alternatively, they might pass between the cells through gaps, and this is the paracellular route. And thirdly, there's the possibility they'll be taken up or phagocytosed by professional phagocytic cells in the specialized cells of the immune system, such as the M cells in the gut. And I want to talk about these three mechanisms in a bit more detail. But one thing to note is that in the gastrointestinal tract, the contents of the lumen do not have unimpeded access to the epithelial surface. The intestine is lined with a layer of mucin, and this has an average, forms a trabecular network with an average effective pore size of between 10 and 500 nanometers. And so that means that it provides a significant impediment to the diffusion of particles from the lumen, which have been ingested, to the epithelial surface before there's even any opportunity for absorptive uptake. And so even small particles which can penetrate the mucin may be impeded by interaction with the macromolecular composition of the mucin through electronic and hydrophobic interactions. And for larger particles, the mucin layer will provide an effective complete barrier through both steric and filtering effects. And so these this delay from the arrival at the potential site of uptake means that the competing effect of gastrointestinal transit can result in the harmless elimination of the particles through the GI tract before they even have the opportunity to be absorbed. So there's not just the physical chemical factors influencing uptake, but the fact there's a competing physiological process to remove the particles from the lumen, which is the natural transit of the contents of the gut through to the anus and fecal elimination. The upper particle size limit for the endocytotic mechanism is about 500 nanometers. So only the small particles can be taken up by this process. So particles between 1 and 10 microns are considered to be taken up by the process of phagocytosis by the specialized M cells or microfold cells, which are present in the Peyer's patches, which are part of the immune surveillance system. And it's hypothesized that the particles then enter the mesenteric network of lymphatic vessels, draining the Peyer's patches, and from there, ultimately could reach the systemic circulation. Now, for larger particles, let's say between 5 and 150 microns, there may be another mechanism by which although they are blocked from the mechanism I've spoken about, they could still enter the circulation. And that is the so-called paracellular route, involving a mechanism which has been termed persorption. And this is the passive absorption of microparticles through gaps in the intestinal mucosa that result from mechanical kneading of single-layered epithelial cells resulting from cell shedding, particularly at desquamation zones of the villi. And this would then result in the particles being removed from the intestinal wall into the lumen of blood and lymphatic vessels. The problem here is that the phenomenon of persorption has not been studied well at all for microplastics. It's been studied for other particle types, and also most of the studies have been conducted some time ago, and we probably could benefit from focusing on this phenomenon in more detail, specifically for microplastics, using standardized methods and materials. But in general, we could conclude that absorption of particles greater than 10 microns is negligible from the GI tract, whereas absorption of those which are less than 10 microns, whilst it can occur, probability increases as the particle decreases in size. When we get to really small particles, nanoparticles, most of these are detected in the mucus lining the intestinal tract, and they don't seem to permeate right through to the epithelium of the GI tract. And so the small intestine seems to provide an effective barrier against nanoparticle uptake, although pathophysiological factors which can compromise the integrity of this lining, for example, impairing the mucus film layer, may result in greater uptake of nanoparticles. And so this is one aspect that leads to some concern that whilst perhaps normal physiology means that exposure to nanoparticles via the gut is minimal, there could be factors which mitigate against this and could lead to increased susceptibility. When we get to disposition, that is, even those particles that are taken up across the GI tract, when they're if they're even quite small, greater than 1.5 microns, they don't appear able to get into the normal capillary of tissues. And so they're not expected to penetrate deeply into the organs. So even although they may get across the GI tract the epithelial cells, the GI tract, they have a very low probability of getting into the tissues themselves. And for particles greater than 20 microns, it appears that any tissue uptake is minimal. But here again, information is lacking. And paradoxically, as Todd has pointed out, whilst we have information on larger particles, it's the smaller particles that most concern is about, because those are the ones where there may be uptake and even distribution into tissues for the submicron particles. And it's those particles where we have almost no information. So once the particles are in the gut, are they eliminated? And a vast majority of elimination studies are done by studying the stool, the fecal material, and particles have been identified. We should recognize that the feces represents a direct route from the mouth out into the environment again, and without any uptake into the body. So that means that the failure to absorb a material from the GI tract will result in its elimination in the feces. So that you cannot relate fecal content of microparticles to systemic exposure, internal exposure. That would need more different type of study. And these studies have not yet been done. So a large amount of what appears in the feces is ingested and excreted directly. But we have to recognize also that there are other routes into the feces which don't result, are not a consequence of oral ingestion. For example, inhalation of material can result in clearance by the mucociliary transport mechanism from the upper airways, followed by swallowing into the GI tract, and that material then ends up in the feces. And this is a normal clearance mechanism of inhaled larger particulate matter. And then for that material that penetrates deeper into the lungs, where there may be clearance by phagocytic processes into the lymphatic system, again, some of that material may end up in the feces. So that even measuring fecal material and trying as a surrogate for oral exposure is not without its pitfalls. And as Todd said, mass balance studies are really important here. Another unknown is how much, if any, of microplastic that is absorbed is ultimately eliminated from the circulation or from the tissues. And there is a possibility that it's phagocytically cleared into the lymphatics, and that ultimately ends up in the GI tract. But this is an unknown area of research. So as Todd has pointed out, microparticles and nanoparticles are complex and varied material. And I've spoken a lot about the influence of size on absorption and distribution in the body. But we need to consider, what is the right dose metric in kinetics? Is it the particle number? Is it the particle mass? Or is it the particle surface area? And going more deeply, we have to consider how some of the other factors that contribute to particle variation might affect their kinetics. So for example, surface charge and other surface characteristics, their shape and the composition. And this has been poorly researched to date. You've heard how important it is to subject studies to QA and QC considerations, and this is equally important for toxicokinetic studies. And as far as I know, we do not yet have a guidance for the screening of such studies as we have for exposure and outcome. And I think that this is just as important and should be looked at in detail. And the same, many of the considerations that one would apply to evaluating a study for exposure or outcome assessment would apply to considering its applicability for assessing toxicokinetics. And so one can envisage a screening and prioritization guidance for studies and evaluating toxicokinetic data, just as there is for other aspects of the risk assessment of micro and nanoparticles. So in terms of key messages, the physiological mechanisms for the uptake, distribution, and elimination of microplastics tend to minimize tissue exposure. The probability of uptake into the body does increase with the decreasing size of the particle, particularly less than 10 microns. There is insufficient information to assess biodistribution, uptake, retention, clearance, rate of translocation, including the likelihood of these particles will cross biological barriers after deposition onto the epithelial tissue or after reaching the circulation. And one of the gaps is that whilst we have an increasing number of publications on the appearance or identification of particles in different tissues, there's much less knowledge of how they might have got there and what would happen to them once they get there. Dosimetry models are needed for extrapolation of the results on particle ingestion obtained in experimental studies in animals to human subjects. And data on the biokinetics of particles obtained in models in vitro cannot be directly extrapolated to the situation in vivo. And we have to accept that some of the models that are being used are not suitable for this purpose. For example, looking at a cell in culture in the absence of a mucin layer does not allow extrapolation directly to the likely uptake in humans where there is an intact mucin layer. And so finally, in terms of research needs in the area of toxicokinetics, I think we do need information on the absorption, uptake into the systemic circulation, distribution and elimination of particles of a variety of sizes on a wider range of plastic polymers and types of particle. And we need a comparison with particles which do not, with a non-plastic composition to determine how much of the information on other types of particle can be applied to those for microplastics. Appropriate model systems need to be used to obtain this information. And more research is needed, as you've already heard, on the influence of the food matrix on the bioavailability and accessibility of ingested particles and the efficiency of the absorption and elimination from the GI tract. Thank you.
Thank you very much, Professor Boobis, for this very clear presentation. And I love the figure where you present the particle characteristics. Very simple and showing that we have to take a lot of things into account. Thank you. Moving to the third speaker of this webinar, being Dr. Stephanie Wright, who is a lecturer at the Medical Research Council, Center of Environmental Health, also in Imperial College London. Her research focus is on microplastics, so definitely an expert in the field, especially in exposure and biological impact, which began with her PhD at the University of Exeter. I'm not sure that's the right pronunciation, sorry for that. She then joined King's College London and held 2 fellowships, moving into the Imperial College of London. And now she is leading the microplastic team, who is an interdisciplinary researcher addressing the issues of human exposure to microplastics, especially quantifying airborne microplastics in health-relevant size fractions, developing techniques for detection of microplastics in complex environments and biological matrices, and also assessing the toxicology of toxicity of microplastics using humans in vitro models. Over to you, it's Stephanie.
Thank you. Sorry for giving you such a wordy bio.
You should be proud of it.
I also realised I just used the WHO report title and should have probably used something more specific, but there you go. So Okay, I guess I want to start just by framing this chapter. We know from decades of research in other particle domains, such as occupational epidemiology, coal, silica, asbestos, air pollution and engineered nanomaterials, that exposure to elevated concentrations of particles can be implicated in disease. And we also know that the toxicity that triggers this is in part governed by particle properties, or what we call physicochemical properties of the particles, with some being more toxic than others. And by physicochemical properties, what we really mean are physical and chemical. So some physical properties would be things like the size of the particle or the shape, and chemical would refer to things like the bulk composition of the material, surface reactivity, perhaps surface charge. And when we talk about hazards, or specifically hazard characterization and toxicology, what we're really interested in is the relation between the dose of the substance or material of interest and the biological response. And often that biological response is considered an adverse effect. So that's just to set the scene here. Just trying to move my slides. Oh, there we go, too far. Okay, so how did we approach our toxicology chapter? Because clearly there's a lot of research going on. It's increasing exponentially and it's increasingly difficult to keep on top of it. So we tried to be systematic and transparent with our approach. So we performed a literature search, which was fairly simple. It just included microplastic and toxicity or synthetic fiber and toxicity. And we searched PubMed as our database. And then we also hand searched some key reports and reviews for extra references we might have missed. And we acknowledge this might not be fully comprehensive, but we still retrieved a lot of studies this way. And then we had an additional part to this literature search and review. We then incorporated a quality screen. So essentially, this was a way of assessing how fit for purpose the studies were in respect to human health risk assessment, which was really sort of our overarching aim as to whether it was or wasn't possible for microplastics. And so in order to do this quality screen, again, we wanted something transparent and systematic and fairly standardized. So we developed a nano microplastic toxicity study assessment tool to do this. So this tool, as sort of both Todd and Alan have mentioned now, essentially scored studies against 3 key areas. And so each key area had a range of different criteria which the study was scored for. So this largely encompassed particle characterization, so the challenge or the stressor or what you're introducing into your system, the experimental study design, so then how you actually approached it and all the methodology and interpretation there, and then the applicability for risk assessment, which included things like, importantly, whether there was a dose response relationship, a concentration range, or an effect threshold established. And this is something that we've mentioned has been published by Todd and a few other authors in Microplastics and Nanoplastics journal. So importantly as well, this tool was really developed. It sort of took attributes from a few key tools that already existed, notably the Klimisch reliability code, the ToxR tool, which is a toxicology data reliability assessment tool, and then the Guide Nano tool, which adapted the ToxR tool for nanomaterials, and then we further adapted it specifically for thinking of plastics, micro and nanoplastics. So I think I mentioned the sort of the reason we use this tool in our literature search and our methods was to provide a standard procedure to evaluate and score the quality of studies and do this in a transparent way. So really it's to identify and prioritize studies that were fit for purpose for risk assessment. But equally, it can also help in providing guidance for how future studies could improve their quality and appropriateness for inclusion and risk assessment. And I think it's really important just to highlight that the scores are not value judgments. It was just merely a guide for systematically screening and prioritizing studies for further interpretation. So just to give you an overview of the results, so this is really looking at the demographics, if you like, of the papers we retrieved. So the method found 109 studies. The majority of those were in vivo or in animal studies, with 37 being in animal-free experiments that would have used well, cell culture models. And I think what's important to note here is you can see straight away that the vast majority of the studies involved were testing or looking at the effects of polystyrene. And of course, that is mainly polystyrene beads. So that was 67% of the in vivo studies and 80% of the in vitro studies. And of those 80% in vitro studies, over half of those were investigating the effects of nano polystyrene beads or nano-sized polystyrene. So there were occasionally the odd study that looked at a different polymer. Those are just detailed along the bottom, but really it was just one or two studies for each of those polymer types. So they were considered quite rare. Okay, so I'm going to start first just running through some of the effects that were observed in the in vitro studies. So these are the animal-free studies looking at cell culture models. Most of these studies focused on a model cell line called CACO2. This is a colorectal cell line, and I just following on from what Alan said about toxicokinetics, when it's used in isolation or on its own, it doesn't secrete mucus. So when we think about the toxicokinetics in relation to effects, there isn't that mucus barrier there, and we're really looking at the direct effect of the particles, assuming they have already reached the cells. So the range of effects varied. So there were studies that found no effects, some that observed effects on the metabolic activity of the cell. Some studies found that exposure to polystyrene triggered reactive oxygen species generation. Some studies found that through, I think, gene expression pathways related to membrane integrity, suggesting that there may be effect on membrane integrity as well. And inflammatory and immune pathway activation were observed. And so what I think really it's fair to say that these are all what we would consider sort of early or upstream key biological events. If they were to persist, or if the exposure was to persist and these were to sort of continue and occur, then this could lead to tissue level damage, but they're what we would consider early events, and that's generally what we would look at in cells anyway. As Todd mentioned, it's more for looking at mechanistic pathways and effects. So there were some caveats to these studies. They did all use very high concentrations, and just by way of an example, for one of the papers, I just sort of did a back of the envelope calculation to convert the mass concentration, so the micrograms per mil, into a particle per mil concentration, using information on the particle size and the material density, and they were in the order of, well, as you can see there, it's just short of 1 billion to over 10 billion particles per mil. So quite high doses. Again, most of the studies did focus on polystyrene beads, and over half of these were nano-sized. And of course, what's important are the particle properties, and not all of these actually did what we would consider a very robust or full comprehensive characterization of the properties of those particles that we used in the system. But some key findings from these really in terms of what was driving these toxic effects. So size was found to be an important property. Surface chemistry, particularly aminated polystyrene, was found to be particularly toxic, and exposure duration was obviously an important metric in this, and then shape as well was found to be important. And actually one study which observed the effects of polyethylene fragments versus perfect spheres, pristine spheres, found the fragments with the sort of heterogeneous shape and rough surface to be pro-inflammatory, whereas there was very little effect observed for the pristine spheres. So shape is considered important as well. So just to recap on the quality scores, however, so no studies scored full marks, and just over half of the studies were sort of scoring around half of the total marks available. And what you can really see here is that the green bars are quite quite small. This is in relation to particle characterization. And so I think moving forward, there needs to be better characterization in studies, so we really understand what we're testing or observing the effect of. Is it of charge? Is it of a leaching monomer? Is it of an impurity? Is it actually shape, size, etc.? And the reason, okay, so I'll just quickly move on to the effects observed in vivo studies. First of all, just starting with effects in the gut. So these were all largely mice and rat studies. And so following exposure via ingestion, some of the effects included inflammation of the gastrointestinal lining, reduced mucus secretion from the intestinal lining, and even altered microbiota assembly, which we would call dysbiosis, and an alteration in the function of the gut microbiota as well. Beyond the gut, effects have been observed in the liver following ingestion. So this includes mainly changes in lipids, the fat profile and composition, or metabolic disruption as well. And then of course, there are still studies that following ingestion find no effects. So one thing I want to highlight about a lot of these studies is, again, these studies in animals all mostly used polystyrene as a polymer. Here's just a handful of studies as an example. This isn't all of the studies from the paper. You can see the doses again are quite high, but I think one of the key things is that a lot of studies administered the dose via water. The animals had ad hoc access to the water, and so the true dose which the animal was actually given is uncertain, is unknown. So to really understand a dose in relation to a response is quite challenging from those studies. So further, moving on from the sort of direct effects, if you like, in the digestive tract and related organs, there were also studies that then followed on looking at reproductive or multi-generational effects. So for example, pregnant mice who were exposed to polystyrene beads, there was actually effects observed in their offspring, but also in their offspring's offspring when the offspring was mated with an untreated male. So we call that the F2 generation. And so metabolic disruption or disorders were observed in offspring down to the F2 generation. And an additional study has also been, again, this fatty acid metabolism disorder observed in F1. So it does look like effects are observed in offspring. There is no effect on sex ratio, survival, body weight, and liver to body weight ratio in the offspring, however. And again, it's really important just to flag that there is this question about dosimetry. So if it's administered via water, what is the true dose that the animal is receiving, since they have free access to said water and may consume it at different rates at different times. Equally, whether this effect is particle or chemical. So is this due to a attribute of the physical particle itself or its chemical properties, or is this actually linked to something else like a leaching monomer? Are there unknown additives present in the material that are leaching out and actually driving these effects? And I think these are some of the things we really need to consider. There are other reproductive effects observed. This is considering the reproductive system rather than generations and offspring. These involve things like the effect on the number of viable sperm and sperm deformities, and inflammation as well. And again, with all of this, there's still the same caveats in whether these are due to the particles themselves and something about the plastic or the bulk composition or the chemistry of that polymer, or if it's actually because of an additive or an impurity that's present and is leaching out and driving these effects. Another thing which came out of this was the recommendation really to use standard test guidelines. Often studies are approaching these using different time durations, different concentrations, dose ranges, and really some standardization will help to sort of be able to draw patterns across the different studies. And this is something that's been recently discussed and covered in a paper published by Scott Coffin in working towards a framework for microplastics in drinking water in relation to health. So when we look at the quality scores for the in vivo studies, again, you can see as a bit of a range. None of them are scoring too high, and the fall down again does seem to be on the particle characterization side of things. And I think that's something as a field we can discuss further and really sort of agree on sort of minimum measurements and reporting criteria. So just to summarize the key messages from the report, a lot of the hazard and tox data is limited to monodispersed polystyrene beads. And whilst these might be useful for asking very sort of specific controlled questions about certain physicochemical properties, it's very difficult to extrapolate the observations to what we experience every day, being that heterogeneous mix of weathered microplastic material. And moreover, what we know in terms of exposure, and especially the size distribution of microplastics we are exposed to, is that they're generally quite, those particles are in the sort of coarser size ranges, whereas a lot of the tox studies have focused on small, nano-sized particles or very small micron-sized particles. And so we've just got a bit of a gap, the sort of area in between the exposure measurements and the tox experiments. But so far there are some suggestions that nano and microplastics might have similar adverse effects to other solid insoluble particles, which could be through similar modes of action. So that would be things like triggering oxidative stress or inflammation and affecting metabolic activity. Essentially, though, it was agreed that at present the data is still insufficient in whether we can draw a conclusion on either side, and so that really we can't say if microplastics are linked to any direct pathologies or not. So just because I'm running over, I'm really sorry about that. So I'll quickly run through our research needs. So I think the common theme throughout has been understanding the sort of not just the mixture that we're exposed to, but then the complexity of the particles themselves, and understanding the effect of different attributes such as size and shape and the type of material it is. And linked to that is then this better need for particle characterization, especially as we're kind of all going off now as researchers and making our own particles to address this first point, but actually if we're all doing this in a different way with a different machine or a different method, who knows what types of artifacts or background contamination we're introducing and impurities. And so I think that's something we need to be really sort of hyper aware of. Dose response, I think we definitely need to be working towards dose response, and we want to be able to compare the potency of different particles. So we really want to have some toxicity values like effective concentration 10 or 50, so we can draw comparisons quite neatly across different particles or different characteristics. There's definitely a need to incorporate standard test methods, again for consistency and sort of data quality. Dosimetry in test systems, this is something that we considered to be a challenge as well. I mean, we kind of mentioned dosimetry throughout this talk in relation to dosing animals via water. I think there's an equal challenge in vitro in cell culture systems where you might have different densities of polymers and particles that are settling at different times, and so the actual delivered dose to your cells might be different for different particles, and I think that's something that we've not really addressed much up to date. Finally, extrapolation is obviously a challenge, as mentioned, not just because of the complexity of microplastic in our environment, but because we don't also have the tools. So when we think about in vivo animal experiments and the extrapolation of that data to in vivo humans, it's we don't have the specific models to do that yet for nano and microplastics. And then finally, this is just from me, I think what a really important thing moving forward is considering microplastic in relation to all the other particles we're exposed to, and sort of understanding where they fall. And I'm really sorry for rushing through that. I just like Todd and Alan, I just want to say thanks to everyone involved in the report, because it really was a team effort. So thanks to all the co-authors and everyone else who's had a part to play. And thank you for the chance to present today. And sorry, it was a bit of a rush, but thank you very much.
Thank you very much, Stephanie, and you did an excellent job. Thank you. We didn't feel rushed at all. It's great that you gave us such a nice overview of the report.
A lot to
and
get
indeed,
through.
Indeed, it's dense information. So I wish to thank you all the speakers for this contribution. And now here my job is done, because I will now hand over the words to our co-host, John Normal, who will give us small recommendations, an overview and a wrap up. And also he will be animating the Q&A. So don't hesitate to put any Q&A in the Q&A chat box for the panel and also for Norm. Over to you, Norm.
Thanks, Isabel. So I just have a couple of slides to wrap up the WHO recommendations, specifically on what's needed and necessary to advance the field and develop research necessary for risk assessment. As you've heard from our speakers today, the purpose of this report was to assess the exposure and effects data on microplastics and to evaluate whether or not a risk assessment could be conducted. We've also heard from our speakers today that there were a number of shortcomings with the data that is currently available, that the WHO concluded that they were not able to conduct a robust risk assessment on health or environmental effects. And so with this conclusion, they came to a number of recommendations for researchers to develop data to assist organizations and regulators to conduct this risk assessment. Really, this WHO recommendation is a call to researchers from all sectors to collaborate and develop this data necessary to conduct a risk assessment. And so just very quickly, one of the first research needs that the WHO noted was the need for the development of standard methods and materials. They specifically called on developing robust QA/QC-able methods that use suitable reference standards that are specifically representative of environmentally relevant microplastics. Particle characterization is extremely important. Stephanie just described, there are different properties of microplastic particles that have different effects in the in vitro and in vivo systems. And so it's very important to be able to identify and characterize size, shape, and composition of these particles, and to also conduct environmental monitoring studies to be able to characterize what's actually out in the environment. This information will help inform human health exposure and help inform the development of reference standards for materials for toxicity testing. It's also important for us to get a better handle on the sources of microplastics and to be able to identify where the microplastics are potentially coming into the environment. And this will help future abilities to help mitigate microplastic into the environment where risk is actually identified. Next slide. Uptake and fate of ingested nano microparticles. We need additional absorption, distribution, and elimination information needed on environmentally relevant microplastic particles. And as Alan mentioned, we need to better understand the influence of the food matrix on the bioavailability of ingested particles, and how does this affect the efficiency of their absorption and elimination. Finally, on the toxicology experiments, we need to have QA/QC experiments suitable for risk assessment. One easy place to look for that would be the OECD testing guidelines that have been vetted and used throughout the world, but those are not by any means the only types of studies that we should limit ourselves to, but we do need to look to see what types of studies are needed to inform risk assessment. And then finally, we do need to, again, adequately characterize the most commonly exposed types of microplastic particles. You may have heard throughout the discussion that a lot of the studies have been done in polystyrene beads. And so we need to be able to use additional materials that people are exposed to out in the environment in order to get a better handle on the potential risks. And so with that, I'm going to close the Q&A session. Please feel free to reach out to Isabel or myself. Our emails are at the beginning of this presentation. That will be posted on both ACC's and ILSI's website. I want to thank our speakers once again. I want to thank our co-host, ILSI, for helping to put together this webinar. And then I want to thank all of you for taking the time out of your day and attending this webinar. Thank you all very much and have a great day.